Hi, this is the conjunction junction team. Did you eat the avocado?
I did. I made guacamole yesterday.
Yes. Okay, so we are discussing how do you live? I love it. Chapter 4, membrane structure and function. Reminder that we gave the quiz a little DOS boot. It is not tomorrow, Thursday. We are doing the quiz Monday the 23rd. Okay? So if I was going to get some key things here about a membrane, first thing I'd want to know is what are the parts of the membrane. And you want to know what each of those things do. So a big component of the membrane would be the phospholipid bilayer, right? And we want to know the key thing on the phospholipid bilayer is the heads are the hydrophilic part, the part towards the water, right? And then the legs in the middle, those are hydrophobic. And this is like the consistency of what? What did I tell you? Olive oil. And if you're a plant, you have a what that goes around the outside? Cell wall to kind of hold it together, but we don't. So what's something in here that can help with the fluidity and maintaining so it doesn't get too hot and break apart and doesn't freeze?
Cholesterol.
Cholesterol. And if I was going to describe cholesterol, I know it is a lipid, in particular a what? Starts with an S? Steroid. And we know that steroids, because we already learned this, are made out of 4 carbon rings. Okay. Okay. So if I look, see? 1, 2, 3, 4 carbon rings. See it? See it now? All right. Then the other big part of cell membranes, or what goes across these, are proteins. And we know the functions of proteins. Let's do them together. Ready? Here we go. Here we go. They could be channels, carriers, cell recognition, receptors, enzymatic, or cell junctions. Good. So that's the role of proteins. Then to the outside, and membranes don't look the same on the inside and the outside, right? Because they have something attached to them on the outside. Those carbohydrate side chains, collectively called the glycocalyx, but if they are attached to a protein, they will be called a glycoprotein, and if they're attached to a lipid, they will be called a glycolipid. So don't let that confuse you, okay? Because they are not lipids and they are not proteins. They're just attached to lipids and proteins, right? And it's that carbohydrate side chain, and we looked at one of the functions is that helps us to know when we're crowded, cell recognition, knowing cell from non-self are all part of those carbohydrate side chains. If we look to the interior of the cell membrane, we could see what? What is this a part? These little purpley things. Cytoskeleton is help support that. So we know each of the components. We know the phospholipid bilayer is a barrier. We know the multiple functions of proteins. We know the function of cholesterol with stability. And we know those carbohydrate side chains with cell recognition. Okay, so if we looked at an unlabeled picture, you could label it, right? Okay. I'm going to jam through here. I'm going to jam all the way through here. And I'm going to go all the way to here. Okay? So the next thing I want to talk to you about is how things can cross the cell membrane. And remember, there's 3 big ways that the items can cross the cell membrane, right? If you want it to cross. One way is what? Through the, do it with me, through the phospholipid bilayer, but you can't do that if you are large or charged. So if you can't get in that way, maybe you could use a channel or a carrier. Now, if you use a channel or a carrier and you go with the concentration gradient, going from a higher concentration to a lower concentration, and you're using a channel or a carrier, that would be called?
Passive.
It would be passive. Another name? What kind of diffusion? Because I'm using a channel or a carrier. Facilitated. Who's doing the facilitation? The channel or the carrier. Now, if I use the channel, not the channel, but if I use the carrier to go against a concentration gradient, so I bind with something and bring it from where it's less concentrated to where it's more concentrated, I'm trying to get it to one side of the membrane to serve a purpose, then that's not called passive transport, that's called active transport. What is our most famous active transport protein? It's called the sodium potassium pump. 3 sodiums out, 2 potassiums in, and it costs you ATP to do that. That's why it's active transport. Okay, so let's review so far. How do we get across? Through the phospholipid bilayer, but not if we are large or charged. Okay, Option 2, use a channel and a carrier, and that could be both passive or active. The third way to get across the membrane is whole membrane, doing what? Endo or exocytosis. Now, when I think endocytosis, I know 3 different types of endocytosis, don't I? What's the first one? Cell drinking. What will we call that? Close. Pinocytosis. Think. But don't. Okay. And then the second way would be what? Phagocytosis, which is cellular eating, like a white blood cell eating bacteria. Okay. And the third way, ding, 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 ding. Receptor-mediated endocytosis. Is endocytosis and exocytosis going to cost you energy? Yes. You're moving your cytoskeleton, which moves your cell membrane, so yes. When you move your skeleton, it costs you energy. So you have to move your cytoskeleton to do that. So now, we understand permeability. We understand this, right? Yeah. So diffusion is anytime you go diffusion is anytime you go from what? Higher concentration to a lower concentration is diffusion. And if you use a channel or a carrier, then it's facilitated. If you go against the concentration, it's active transport. Now, we can look at things that can move through the phospholipid bilayer. Things like carbon dioxide can move through. Oxygen can move through. Water can move through. But water can have some special proteins that allow it to move really quickly through. What's it called? What's water's special protein? Aquaporin, baby. Okay? Because I'm pouring some water, pouring some aqua. Okay. But when we talk about water moving across a semi-permeable membrane from a higher concentration to a lower concentration, that has a special name. What is that special name? It's a term. Osmosis. Okay? Osmosis is the diffusion of water through a semi-permeable membrane going from a higher concentration to a lower concentration. Right? So that's just you going right through that phospholipid bilayer. And we saw several incidences of that. Okay? But one that we could do is we could do this tube right here. Okay? And we have words to describe. It's always one relative to another. You wouldn't say, I'm left. You would say, I'm left of this individual, or I'm right of this individual. You don't get to say I'm up unless there is a down. It's always relative to something else. So the way you describe solutions is water, we know this, water must flow, what? From the hypo, and it's going through the membrane into the hypertonic environment. So to identify which environment is which, or which solution is which, whoever has the most stuff in it, we say that is the what? Hypertonic. So this side right here is hypertonic, right? Or it was over here. Hypertonic, because it has all of these osmotically active substances in it. So if this is the hypertonic side, because it has more stuff, then this side is the hypotonic. That's all it can be. It's one relative to the other. So the hypertonic side has a lot of stuff in it, a lot of osmotically active substances in it, so it has less water. So water is always more concentrated on the hypo side. And water must flow from the hypo. So it's going from where it's more concentrated to where it's less concentrated. So whatever side is hypertonic, is this solution is going to get bigger. It's going to increase in volume because water is moving in. So if this were a cell right here and water was moving into the cell, this cell could expand, right? And if this one is expanding, then this one is getting what? Smaller. It's always one relative to another. And if you remember, we talked about, okay, if I went like this and I put a solution in here, and on this side I put a bunch of stuff, okay, and this and this is side A, B, and C, right? Then C relative to B is what? it's hypo, and B relative to C is what? Hyper. But B relative to A is hypo, and A relative to B is hyper. It's always one relative to another, right? So now describe, oh my gosh, it's so messy, I'm embarrassed you're taking a picture of it. If I take and cut myself and put ginormous red blood cells in here, okay, let's all talk about how the red blood cell is one to the other. Let's assume one of these is isotonic. What does that mean? Same, right? So the amount of water that moves in is equal to the amount of water that moves out. Okay? So whisper back and forth with your bio buddy. I want to give you a minute to think about it. Which solution would be hypertonic to the red blood cell, hypotonic to the red blood cell, or isotonic to the red blood cell? Because we're going to label it right here, what it is. Okay, what do you want to call this one right here to the red blood cell? Hyper. You're saying it's hypertonic. This one is isotonic, and this one is hypotonic. So if this solution is hypotonic, that means the cell is hypertonic. So water must flow from the hypo, so it's going to flow in to our red blood cell. Now, is there some still moving out? Yes, but more is moving in than out. What could happen to our red blood cell? Burst. Would that happen to a plant cell? No. Why? They have a cell wall. What about in this isotonic situation?
Stay the same.
Right, so the same amount of water that moves in is moving out. Same. Okay, for us, that would be a 0.9% salt solution if we were talking about for us. Okay, in this situation, if it's hypertonic, water must flow from the hypo. So what's going to happen? It's going to shrink because water is going to flow out. Is any water going to be flowing in? Yes, but not as much as flowing out. So this cell will shrink. Would a plant cell shrink? No, why wouldn't a plant cell shrink? Cell wall. Could it pull away from the cell wall? Yeah, what's that called? Plasmolysis. Do you remember that when it pulled away? And when this shrunk, then it, what do you remember what that word is called when it shrinks? Crenates. Remember that? Like that's the name of it. And when a cell swells, if it's a plant cell, the plant cell wall exerts turgor pressure and pushes back. Do you remember that part?
Yeah.
Okay. I have a cucumber. Here is my cucumber. Those are the bumps on the cucumber.
Looks like a table.
Okay. I have a cucumber. I want to turn this cucumber into a pickle. What solution do I need to put my cucumber in to turn it into a pickle? I need to put it in the hypertonic solution. So that hypertonic solution that hypertonic solution causes it to shrink, right? Because water leaves it and it gets smaller. I have some carrots. And they're kind of ugly. And they're kind of wilty, but I'm having a dinner party with guacamole and chips and some ranch dressing to put my carrots in, but my carrots are a little wilty. I want to firm them up. I want to firm those carrots up so they're crunchy. Where could I put them? Yeah, if I put these carrots in distilled water, oh, okay. If I put them in distilled water, water would flow into them and I would have crunchy carrots again, right? Okay, good. So when we look at this, are we feeling comfortable with this? These are the three scenarios we created. All right with that? Just take a picture now. Okay, so when we look at this, this is a red blood cell and a plant cell in a hypotonic solution. It's swelling out. Yes?
Can you go back to the previous picture before?
Yes, this one?
Yeah.
There you go, my dear.
Thank you.
Again? Okay. So this is in a hypotonic solution. It's swelling, right? And we talked about when it swells like that, it can burst. That's lysis. And in here, the cell wall pushes back. That's turgor pressure. Turgor pressure. And this is lysis. Or cytolysis. Cyte is cell. Lysis. Okay? And in scenario number 2 that we discussed, this is when you put a red blood cell in a hypertonic solution. It's shriveling. What do we call that? Crenation. And we can see the cell membrane is pulled away from the cell wall. Okay, so crenation is when it shrivels. Plasmolysis is when the cell membrane pulls away from the cell wall. And we put that all on our chart, right? And then here we have it in an isotonic solution. So the same amount of water going in is going out. No net change. Right? We talked about using a protein. Every time you use a protein, it could be a channel or a carrier. If it's passively using it with the concentration gradient, what do we call that? You already told me. Starts with an F. Facilitated diffusion. Good. Okay. If we have to use energy, it is active transport. Okay. Then we talked about endo and exocytosis. If you see it grabbing something and bringing it in, it is endocytosis. If it grabs liquid, it's called what? Penocytosis. If it grabs food, it's called phagocytosis. If a bunch of things bind, ding, 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 and then goes in, it's receptor mediated. When it takes a vesicle and it's docking, and ends in release, that's exocytosis. If it ends with it being inside, it's endocytosis, right? What's your ending spot? Oh, I'm inside. It must have been endocytosis. What's your ending spot? Oh, I'm leaving. Must be exocytosis. Look where it's ending up. Okay, so this right here is what again? Phagocytosis, cellular eating, cellular drinking, and then this was receptor-mediated endocytosis. And then here, I think I have some questions for you. I think I do. I'm going to have to remove some students. Hang on. You're going to be your favorite party food because we were talking about carrots and ranch dressing and guacamole. I love party food. It's so bad for you, but I love it so much. Tostitos.
Oh my god.
Water? That is so not a fun party food. Any food. Lay's potato chip. Do you like sour cream and onion? My Lay's potato.
No.
Or the barbecue? No? What one do you like?
Just like regular.
Oh, just plain Lay's?
I like just
Like, how about regular Lay's in onion dip?
Oh, that's good.
No?
That's good.
Ruffles? Do you like artichoke dip and use
Yes.
the tortilla
Yeah.
chips or like
That's
the little
good.
dry pieces of bread?
That's so weird.
Okay, here we go. Got 4 questions.
Yeah, we can. Yeah.
I'm going to show the questions. Showing you the questions. I'm showing you the questions. I'm showing you the questions. Do you feel smart? Are you glad you came in for the review today?
Yes.
All right.
I think we did these. I think we did. I was watching this. Look, I can just take a picture of it and then you post it. You're in danger. You're in danger. Oh, you just like screenshots? Yeah, my phone also no, yeah.
We ready? Are we done? Okay. Yay! I'm so proud of you. Yay! that's 2. I'm so proud of you.
Another one.
All of these are endocytosis. Yay! Yay! Good job, you guys. I'm so proud. Oh, sorry, we have 8. Do you want to do the other questions?
Yeah.
Okay, let's do them. Let's do them.
Smarter than me.
Let me find. Come on now.
Here.
We could. Do you want to do the interactive ones together,
Here, I got 4. I got it.
or no? It's up to you. This is a monster one.
Oh, I know.
How many teams do you want to be in? 3. 3? Okay. Done. Are you ready? Go.
Yeah.
I'm going to pause this because they don't know what's going on. Okay, so good job, Snowblinders. Let's review our questions. Oh, I've never seen that monster before. It's number 1. All right. Phospholipids have a hydrophilic head and a hydrophobic tail. Only a third of you got that. A bunch of you said a hydrophilic head and a hydrophilic tail. They're not both philic, remember? Right? You're imagining the ball with the 2 legs, the hydrophilic head, hydrophobic tail. Do you know what I'm talking about?
Yeah.
Okay. And then cholesterol, hey guys, cholesterol is found in, no. Oh, choose the false statement. I'm all, that's not true. That is, they're not found in all cell membranes. Where are they found? Animal cells. Good. The extracellular matrix functions in support and communication. That is true. Identify the false statement. The inside and the outside are supported by the cytoskeleton. No, only the inside. Good. And identify the false statement. The carbohydrates are not the same because remember that helps us identify self from non-self and also who you are. It's by your extracellular matrix. And that is it on that. Okay, let's go to the next. Donde está? Here we go. We'll just do this one together. Okay?
Teamwork.
Teamwork. All right.
Oh, junction joins cells.
Okay, there we got one. Junction joins cells.
Cell recognition.
Okay, wait, don't do them all. So let's do carrier. Do you remember what one is carrier? Interacts with specific molecules, self or non-self, metabolic function, substances bind to it, molecules cross freely. No. Substances what? Bind to it? There we go. No. Interacts with specific molecules. There you go. What's a channel? Molecules cross freely. Good. Cell recognition. Cell from non-self. Good. Receptor. Substances bind to it. Enzymatic. Metabolic function.
I can't even.
When you review it, can you see them a lot bigger? No, you can't.
I cannot see.
Can you
Okay. Yes.
Can you like
do like this?
Yes, I can. Yes, I can. What are you?
Yeah,
I knew what you meant. You're like, can you make it bigger?
Awesome.
I don't know that you can see it all that well.
It's like blurry. I still
Yeah.
can't see it.
It's fine.
I'm sorry.
That's okay.
Okay, the next, oh, here's one. Ooh, 5 questions, I'll pause it. Resume. Okay, here we go. In. Yay, wait, that is 5. That is 5. All right, so cholesterol is found in the plasma membranes of animal cells. Somebody said plant. Get it in your mind. Animals, cholesterol. Animals, cholesterol. Why don't plants need it? They have a cell wall. What can't pass through a phospholipid bilayer? Large molecules, large in charge. Oh my gosh, we could have had like so many. Passive transport does not require the input of energy. Good job. Active transport requires energy. It goes against concentration gradient. Excellent. Let's see if I have another one. Yes, I do. Yes, I do. And then I think you've done them all. Here you go. I will pause again. Okay, here we go. How did we do? Oh, water is always going to be the solvent. Whatever you dissolve in it is the solute. Okay? Solvent, solute. Put away now. It's even neater. Okay. Next, the hypotonic solution contains the most solutes. No. Remember, the least. Okay, so that's worrisome to me. Remember when we did the pictures of A, B, and C and all the stuff in it? I maybe should have reviewed that with you. Whatever one is hypertonic has a lot of solutes. Whichever one is hypotonic has a lot of solvent. Okay, relative to the other. When a cell is placed in a hypertonic solution, think about it. That's how you pickle things. You put it in a hypertonic solution, water comes out of it. Okay, so the water exits the cell towards a higher solute concentration. We had one person say water enters the cell and solute exits the cell. No, it's all about the water movement. And that was it on that one. And we did the last one, so you should be all studied up. Are you happy with yourself? Because I am super happy with you. Good job. When is this quiz?
23rd.
Monday.
23rd, Monday, and you're going to be all studied up. It makes a better weekend. You're welcome, you guys. Make good choices, and if you get hungry, have a piece of toast.